Maintaining sample health is paramount to ensuring the validity of any live imaging experiment. As any exposure to light has the potential to perturb living systems, there is a limited trade-off between spatial resolution and acquisition speed to ensure that your specimen remains healthy. This fact has implications not only for experimental setup and execution, but also when it comes to the selection of the right confocal system to perform high-resolution live cell imaging. To address the challenges of live cell imaging, STELLARIS has been designed to boost efficiency of photon detection while enabling high spatiotemporal resolution with minimal sample damage. To perform exceptionally fast confocal imaging, STELLARIS combines a high-speed resonant scanner with the sensitive power high-D detector family and rapid sampling electronics. The combination of the white light laser with the fast resonant scanner provides two benefits. First, you can study dynamic events even in large volumes without compromising spatial resolution. And the second benefit is that you can image for longer periods, keeping the laser intensity low to preserve your sample from photo damage and phototoxicity. However, the high acquisition speed comes with a price, and this is the reduced signal-to-noise ratio. But this limitation can be overcome by adding the new Dynamic Signal Enhancement, also called DSE, to the process. Then you can achieve the optimal image quality at the required temporal resolution. DSE acts as a rolling average, or accumulation, which for each frame takes also into account the neighboring ones acquired before and after. And this also works on the life image as you can see it in the live view. Because of its rolling nature, you can still acquire at high temporal rates without distorting the temporal characteristics of your sample. This can also be applied as a post-processing step, significantly improving the signal-to-noise ratio. And you can push the limits of resolution and signal-to-noise ratio even further by combining DSE with lightning adaptive deconvolution. You can easily notice that lysosomes which could not be separated from the raw data or from DSE alone are now distinctly separated. Here is a stunning example proving the higher value brought by this dynamic duo. With STELLARIS, you can maintain a superb image quality even when you use very high acquisition speeds of several volumes per second. This is achieved thanks to the powerful trio of the fast resonant scanner, dynamic signal enhancement, and lightning. Imagine performing your research with the best balancing of speed, image quality, and sample preservation. Imagine the power to see more.